I would like to introduce to the stage the next panel, which is going to be on liquid biopsy for brain tumors, led by Dr. Stephen Bagley from the University of Pennsylvania. As you've already heard this morning, this is a very hot topic, and I'm really excited to hear what the panelists have to say. Thank you.
Thanks so much, Lauren. It's a real pleasure to be here. My name is Stephen Bagley. I'm a neuro-oncologist at the University of Pennsylvania. And I am honored to be joined by really a fantastic panel of experts in this area, spanning everything from the preclinical setting all the way through implementation into routine practice. So briefly want to introduce the panelists and then provide a brief overview of the field to kind of get everybody on the same page, and then we'll jump into the panel discussion. So our panelists include Dr. Chen, who's Assistant Professor of Radiation Oncology at Washington University in St. Louis, Dr. Graham Woodward, Professor and Chair of Neurosurgery in the University of Maryland, Lauren Lyman, who is the Executive Director of BloodPAC, Dr. Ying Meng, who's a neurosurgeon and researcher at Sunnybrook Research Institute in Toronto, and Dr. Patrick Wen, who's a Professor of Neurology and Director of the Center for Neuro-Oncology at the Dana-Farber Cancer Institute in Boston. These are my relevant disclosures, including a couple of research relationships with liquid biopsy companies. So I want to provide a brief overview of the urgent and unmet clinical need for liquid biopsy for glioma and other primary brain tumors, touch briefly on this emerging field of FUS for brain tumor liquid biopsies, and then we'll jump into the panel. So as we've heard earlier in the day, this is an incredibly aggressive and difficult disease with a fairly toxic standard of care therapy that, as you can see here, does not improve overall survival by very much. There have been some small advances along the way, but since 2005, we're really still talking about just over a year of median overall survival. Just in terms of nomenclature and what we're going to be focusing on here, so liquid biopsy actually refers to any tumor-derived genetic material that you can detect in a bodily fluid compartment. And so there are multiple analytes one could choose to look at and multiple compartments in which you can look. Today, we're really going to focus on cell-free DNA, both in the plasma and cerebrospinal fluid. There's an abundance of literature, particularly in other solid tumors, but also in the last 5 to 10 years in neuro-oncology, and we'll focus on this for the remainder of today. But I just want to point out that there is a very broad array of other analytes, including EVs, extracellular vesicles, which includes exosomes and microvesicles. There are various cargoes of interest you can look at within these vesicles that include DNA, RNA, microRNAs, and others. And then circulating tumor cells, which are difficult to find, but not impossible to find in primary brain tumors. I think so far in our field, these studies of these other analytes have tended to be single center, small sample sizes, differing methods for isolation, and so I don't want to ignore this. This is a very important topic, just not within the scope for today. So in brain tumors, liquid biopsy in the traditional sense, which means checking somebody's blood, spinning off the cells, looking at the plasma, and trying to detect tumor-derived DNA in that compartment, has been extremely difficult, at least with conventional, commercially available targeted NGS panels. The figure on the left is from a landmark paper from Chetan Betta-Gowda and colleagues in Science Translational Medicine. Can't believe it's nearly 10 years ago at this point. But you can see the very high detection rates. This was using a targeted NGS panel in the plasma across a variety of solid tumors. All the way on the right is glioma. Only 27 patients, but we're talking about a less than 5% detection rate there. Relative to the other cancers was quite poor. And then we actually did a study a couple years ago looking at a commercially available targeted NGS panel. These are basically each column in that figure is a different patient in this cohort, and all of the mutations of interest are on the left. And you can see that the main takeaway here, despite the complex color coding in the figure, is that there really were no concordant mutations detected in tissue and plasma. The two green ones were actually just different variants in the same gene detected, but all of those blues were tissue-only mutations, and all of those red boxes were plasma-only mutations. And upon further inspection of this data, a lot of those red boxes were actually not tumor somatic variants. They were clonal hematopoiesis we were detecting in the plasma. So not performing anywhere where we would need it to be clinically useful. Using CSF might be better. The ctDNA concentrations are much richer in the CSF. There is a paucicellular background, so you get less background noise when you're trying to detect the tumor-derived DNA. This is obviously a more invasive procedure, although much less so than a brain biopsy. So there's a lot of interest in exploring this area as well. And so for the purposes of today, focusing on trying to get ctDNA out of the plasma compartment, FUS-enhanced liquid biopsy, as we'll hear from some of our panelists who have led this work, has shown great promise in the preclinical setting. And this bidirectional flow across the BBB that can be induced by FUS with microbubbles allows us to actually detect these tumor-derived materials that otherwise are not being shed into the systemic circulation. This is some work that I just want to highlight from Dr. Meng, who's one of our panelists today. This was a really beautiful paper where they looked at pre- and post-focused ultrasound plasma samples in human patients. And what you can see here is very clear and consistent increase in the plasma cfDNA levels post versus pre-BBB opening. As we'll talk about, the cfDNA measured here is raw total cfDNA in the plasma. So we actually don't know how much of this is tumor cell derived versus how much is coming from other sources, which may include cells in the tumor microenvironment. It may include, in large part, peripheral immune cells like neutrophils that release their cfDNA into the circulation. But I think that this very robust data showing consistent increases is extremely promising. And then I also want to point out Dr. Hong Chen's recent paper in Theranostics. This is using a pig model. And here you can see the contrast-enhancing tumor volume consistently increases post-FUS, as we would expect. And what was, I think, fantastic here is that they actually looked at a couple of mutations that are characteristic of high-grade gliomas, including EGFR variant 3, which is a very common alteration in the EGFR extracellular domain, and then TERT promoter mutation. And so using a droplet digital PCR assay, they were able to show that detection of this, which is certainly tumor-derived, was higher post-sonobiopsy. So I think really amazing preclinical work. And then I just want to point out also, Dr. Woodward and others are leading these clinical trials that are currently open to accrual. And so I think in the not too distant future, we're going to have some real decent sample sizes of human patients who have been subjected to post-FUS liquid biopsies and start to understand how to use this in practice. All right, so with that, I'll hand it over to each of the panelists to do a brief introduction. And if you can try to keep it to 1 to 2 minutes each, that would be fantastic. Dr. Chen?
Wonderful. I have a slide, one slide.
Yes.
Okay, so thank you so much for the FAST Foundation for giving me this opportunity to join this wonderful panel. I just put together this one slide to summarize our progress so far in developing what we call sonobiopsy, which is I think the focused ultrasound-enabled liquid biopsy is too long a name to call, so we just call it sonobiopsy. So we introduced the concept that the focused ultrasound-induced blood-brain barrier opening enable this two-way trafficking across this blood-brain barrier in 2018, and then show the initial proof of concept using GFP as a marker. And then we move on to show further the feasibility and the safety of this technique in mice, and then recently able to show sonobiopsy can effectively enhance tumor-specific ctDNA release in a pig model of GBM. And then earlier this year, we got IRB approval for our clinical study using a neural navigation-guided ultrasound system. So I also want to take this opportunity to highlight 3 talks that are presented at this symposium. Just to tell everyone, I did watch all the talks. So the first talk I want to highlight is the abstract number is BTU03, so I highly recommend everyone to check this out. So this is presented by the team from Chenggung Memorial Hospital, Taiwan. So they actually showed that sonobiopsy enhanced the concentration of circulating tumor-derived DNA in patients with a recurrent GBM. And then another abstract is from, it's BTU04. It's presented by Cordance Medical about their unique design of their wearable focused ultrasound device and also their design for their sonobiopsy clinical trial. Another, last but not least, BTU07, presented by the team from Georgia Tech. So Kostas and his team show that focused ultrasound enhanced tumor-specific protein secretion to the blood in a mouse model of brain tumor. So I definitely encourage everyone to check out all this wonderful presentation. I'm going to stop here.
Thank you.
Skip that.
Dr. Wen?
Thanks so much for including me in this wonderful panel. You could argue that no other tumor requires liquid biopsy more than brain tumors, given the difficulties of performing these biopsies. And I think the potential for focused ultrasound to make this a reality is really exciting and important. I thought I would just try to highlight some of the potential uses of this next. So I'll talk about the use in diagnosis, in monitoring disease course, and determining molecular targets for therapy over time. And then as technology improves, potentially providing pharmacodynamic effects following treatment. Next. So although MRIs are very helpful in diagnosing brain tumors, there are actually many brain tumors where it's a dilemma before the patient undergoes surgery. The top panel shows a corpus callosal tumor that can look like a glioblastoma, but can also be CNS lymphoma. Below that is a non-enhancing area that could be a low-grade glioma, but in a young patient population can also be confused with multiple sclerosis. And ideally, you would not subject a patient with multiple sclerosis to surgery. On the right-hand side at the top is a single brain metastasis that looks identical to a glioblastoma. And this is a very common dilemma. And then in the pediatric population, one of the deadliest tumors are diffuse intrinsic pontine glioma. And although neurosurgeons are getting better at biopsying this, there's significant morbidity associated with this. And being able to diagnose these tumors non-invasively would potentially be a major advance. Next. Another major area of potential use is tracking disease progression. In 30 to 40% of glioblastoma patients, immediately following their radiochemotherapy, the scans look worse. And it's very difficult to differentiate whether this represents tumor progression or pseudoprogression from the radiation therapy. And if we can measure ctDNA and see if it can predict whether this is tumor progression or pseudoprogression, it would be a major benefit to patients. Next. Increasingly, immunotherapies and viral therapies are also being tested. And in these therapeutic modalities, pseudoprogression is even more common. And so again, ctDNA may potentially have a beneficial role. Next. And then finally, the genotype of these tumors change with time. And so especially with mutations in the epidermal growth factor receptor, about half of these mutations are different at recurrence. And if we have a way to look at the change in genotype over time, this may help select targeted therapies for patients at recurrence and help enrollment into clinical trials. Another issue is that we frequently use the MGMT promoter methylation status to predict sensitivity to chemotherapy. And if there's not enough tissue from the initial biopsy, being able to detect this in the blood would also help us determine whether chemotherapy is helpful. So these are just some examples of the potential roles of the focused ultrasound improving the value of liquid biopsy for these patients. I'll stop there. Thank you.
Thank you, Dr. Wen. Dr. Meng, do you want to go next?
Hi, thank you for the invitation to be part of this panel. Our initial study was done, and thank you, Dr. Bagley, for showing our data, was done in 9 patients with glioblastoma, and they were treated basically in conjunction with adjuvant chemotherapy schedule. So we were excited to demonstrate there was an increase in the ctDNA that we were able to detect before and after FUS consistently with every treatment. And currently our site is part of a multi-center study started by Dr. Woodward, looking at this more in a more controlled way in patients who are newly diagnosed glioblastoma patients going for surgery, so we can actually compare the samples collected after the focused ultrasound treatment with the tissue resected from surgery. So I'm excited to be able to study this question in a more kind of definitive and controlled fashion, and this is really something we're very excited to be part of. Just to add on to, I know we're focusing on ctDNA, just to kind of expand the question onto other disease entities, I think extracellular vesicles are really exciting to look at in blood samples as well. I think, EVs are the way how cells communicate with each other, and particularly in the field of neurodegenerative diseases, it's something that we can look at in our samples after treatment, particularly looking at glio-derived EVs, and that might be able to tell us what the disease stage in these patients are.
Thanks so much. And Dr. Woodward?
Great, thank you. I think that the first step in terms of considering liquid biopsy or sonobiopsy with these focused ultrasound treatments is to get as many samples as possible. I would encourage anybody who's thinking about clinical work or even some preclinical work to be drawing blood or CSF before and after focused ultrasound, whether that focused ultrasound is for mechanical effects or thermobiological effects. I think there's a lot to be understood yet in terms of the biology of these treatments, and we may be able to glean something about what we're doing with focused ultrasound by sampling pre and post treatment. So that was a starting point for us when we began the early clinical trials and thought, we don't really know what we're looking for, but we want to make sure we're studying the potential effects of focused ultrasound in the blood. And so we had in all of our protocols, we sampled the blood before focused ultrasound as a control for that one patient at that cycle of focused ultrasound. And then we also sampled the blood at 30 minutes, 60 minutes, and 24 hours after focused ultrasound. So each patient in all of our studies has 4 blood samples at each cycle, and then it gets processed in a very uniform way. And so that became the sort of foundational bank of samples that we began to look at. And the first step was we started to see differences. Number one, we looked at cell-free DNA, as the Lipsman and Yang had done, and saw very similar results. And then started with my colleague Chetan Bedagauda, who you saw had published the paper in Science Translational Medicine on circulating tumor DNA in other solid tumors, and not found much in glioblastoma. And so his team in the Vogelstein lab at Hopkins are looking at all of our samples, and we're in the process now of validating some of the findings back against the tissue in the patients that had resections, but establishing some form of ground truth with regard to what we're looking at in the blood back to the tumor at the time of sampling is important. And the confounder here for us is that the surgery in these cases was earlier than the focused ultrasound, so there was some time between that surgery and the sampling of the blood or the focused ultrasound treatment, and so the tumor could have evolved somewhat over the course of that interval. So the study that we currently have open, the liquid biopsy or sonobiopsy study, as we should call it, right? So that study does seek to establish that ground truth by performing the focused ultrasound as close as possible to the planned surgery, and then sampling that area that the sonobiopsy was performed in stereotactically during surgery, and then having that match with a very short interval, the tissue and the blood sample. So I think that's where we're going with this. I do think it's an exciting time to potentially enable another method of functional assessment of the tumor. Back to Dr. Frank's question earlier around PET. We want to understand the biology as much as possible of these tumors, because we know the biology is quite heterogeneous. It's spatially localized in different ways in each patient, and we need a closer or highly refined view, not only of structure but of function. And that's how we're going to get closer to where we need to be in terms of designing therapeutics. The problem with glioblastoma, in my opinion, is that we don't understand that relationship, and we don't have a way to pair treatments effectively with the variable functional issues that are highly different in all of these tumors. So liquid or sonobiopsy gives us the opportunity to potentially study function in new ways and understand the tumor, which will get us closer to the point of appropriately pairing treatments with a given patient's tumor.
Thank you. And last but not least, Lauren.
Hi, everyone. Thank you so much for including me today on this incredible panel. BloodPAC is a slightly different organization. I'm the executive director of the BloodPAC Consortium. We're a group of approximately 60 different organizations running kind of spreading the stakeholder field in liquid biopsy, from academics to pharmaceutical companies, diagnostic partners, government agencies, payers, and others. We focus, and our mission really has never changed from our founding as a commitment to the initial White House Cancer Moonshot to accelerate the development, the approval, and the accessibility of liquid biopsy technology for cancer patient benefit. What is interesting, I think, about Focus Ultrasound is we were approached several years ago by the Focus Ultrasound Foundation trying to discuss with us how critical this technology would be in the future for cancer patients and brain cancer specifically. We have been working with them for several years now to try to identify the best ways to collaborate. And I'm excited to announce that Dr. Bagley and Tempest will be leading a newly formed group around CSF-based liquid biopsy for brain tumors, both pediatric and adult. It will be the first working group and project that BloodPAC has specifically taken on that's disease specific. What our goals are for most of our working groups, and specifically this one, will be to focus on kind of standardizing pre-analytical variables for collection of CSF, eventually hopefully blood as well, for the broader community, and then moving forward from there and creating standards for the community as a whole. I think that's kind of our niche. My personal goal and hope for this project, and as we continue to grow, is to start including more of our assay developers in this field. I think typically it has been in underserved fields in general. That's how we actually started the dialogue. What was BloodPAC missing? What were we not addressing scientifically that was deserving of our attention going forward over the next 5 years? And thrilled that we will be commencing this project in the next few weeks. So thank you so much for having me.
Thanks so much, Lauren. So we have a number of, I think, really great burning questions that we thought about as a group prior to this meeting, and I'm going to try to get through as many of them as I can, and I'm going to direct them to specific panelists. I want to start off, Patrick, with you first. So if we imagine a future, hopefully not too long from now, where we've shown that FUS-enhanced liquid biopsy actually leads to pretty high sensitivity and specificity for detecting, tumor somatic mutations in glioblastoma, for example. How do you envision that being integrated into the routine care of patients? At what time points do you think we would be doing this? How often? And what clinical problem do you think is top priority?
So I think throughout the life of the patient, this potentially could be useful. At diagnosis, as I tried to illustrate, having this information would be very valuable. If you can find IDH mutations, you can detect low-grade gliomas. TERT mutations could help detect glioblastomas at diagnosis. I think tracking disease course is really a huge problem in our field, given the limitations of imaging. And so differentiating progression from pseudoprogression is such a major clinical issue right now. And then I think as targeted therapies potentially improve, being able to identify the driver mutations that potentially be targeted would also be helpful. It's given the fact that 50% of EGFR V3 mutations are lost at recurrence, you can't take the patient's initial biopsy and use that as the genotype for guiding treatment at recurrence. And so there are other mutations that also fall into this category. And having the liquid biopsy determine the actual mutations at recurrence, I think would change treatment significantly. So those are just some of the examples, I think.
Yeah, thank you very much, Patrick. Graham, I want to ask you this question. Of all the different liquid biopsy assays or analytes one could look at, what do you see as the highest priority for us in doing these clinical trials? What should we actually be looking at in the blood when we have these precious samples collected pre and post? Should we be looking at total cfDNA concentration? Should we be looking at a targeted NGS panel and if so, which ones? How do you think about that in designing these studies?
I think this field is rapidly advancing, as you know, and I think whether it's with extracellular vesicles or new assays for fragmentomics, these are all things that in the world of liquid biopsy are new terms that are coming out, and people are beginning to understand the correlation between some of these analytes and tumor biology. So I think we as a group, if we're really going to work in parallel with this rapidly advancing field of liquid biopsy out there in the world of solid tumors, we need to stay in touch. We need to be having BloodPack and others come to us and tell us what the most advanced assays are, what's the most promising, and then again correlating that with what we know about tumor biology for brain tumors specifically. So I think that this is a rapidly advancing field, as most of us have seen. It was very telling to me when I was in my residency at Hopkins, when Bert Vogelstein's lab, which was one of the preeminent cancer biology labs in the world, shifted from a therapeutics lab to a diagnostics lab. And they completely, all in, just went in and completely shifted to diagnostics. And that tells you that he is a very smart and insightful individual, and his whole team thought the way we were going to impact the treatment, management, and maybe cure cancer was not by back-end treatments with precision medicine. It was going to be with earlier diagnostics. And so I think we have to follow suit with that in the brain cancer world. I think focused ultrasound may enable that. But again, that world is well ahead. So we need to be partnering with the best and understanding how to use those new assays that they're coming up with to apply it in brain cancer.
I want to ask a follow-up question. So I'm just wondering for Graham and Stephen, so there's CSF-based liquid biopsy, and then I'm just thinking, should we use, based on so many assays that are already being developed for CSF detection, should we use CSF-based liquid biopsy as a benchmark for, I'm going to call it sonobiopsy? Or do you think, I'm just thinking whether we should benchmark with some more sensitive assay or some technology just to demonstrate the advantage of sonobiopsy.
Yeah, I mean, I think that you're bringing up an important point, which is if you're doing any of these studies, what is your comparison or your gold standard? And classically in liquid biopsy, it's tissue sequencing. But what's really created, I think, a complex situation here is that if you sequence the CSF, you often will find mutations that are clearly tumor somatic mutations that you didn't find in the matched tissue. And it may be because of sampling issues or tumor heterogeneity. And so if you do a blood and tissue matched comparison, you actually may not have great sensitivity by conventional metrics, but is that really the right comparison to make, or should we be looking at a combination of tissue plus CSF at sequencing, and that's like your gold standard panel? I think this is an open question and a very important one as we do more and more human studies. It's a great point. Ying, I want to ask you a question about some of the other factors that influence sensitivity, for detection of ctDNA. So opening the blood-brain barrier with FUS is fantastic, but I imagine when we do that, even if it's done optimally, there are many other factors that might dictate which patients have higher versus lower levels, patient-level factors, tumor-level factors. Can you talk a little bit about what are some of the other issues we face with sensitivity for ctDNA detection?
Yeah, for sure. So I guess there are some questions that are still being worked out in terms of the technical aspect of doing focused ultrasound B opening enabled or sonobiopsy as renamed. It would be like the amount of B opening we're doing, does that affect the amount of ctDNA we are able to release? The timing of the blood collection from when the blood-brain barrier was opened, as well as the patient's own, tumor burden in the spots that we are sonicating, whether it's more necrotic versus somewhere that has a greater number of tumor cells. So these are all kind of factors related to the delivery of focused ultrasound that might affect the level of increase you would be getting post sonication. And then after that, collection of the blood is really important. And I think anyone who's worked in liquid biopsy would know that about, when, the sample handling, the amount of time that you are collecting between the blood collection to storing the samples. Those kind of details are actually really important, even down to the type of tubes you're putting your samples in are really important in your ability to detect these markers. Because they're so low in yield to begin with, it's quite important that we are consistent between the different sites that are studying this question.
And as a follow-up to that, there's a question from the audience here that is very related to what you were talking about. So it says, how soon after sonication should the blood sample be taken? What is your answer to that?
I can help to address this question. So this is definitely number one question that we had is when to collect the blood. And we do know the amount of biomarker in the blood that we can collect depends on how much we release, and also there's a fast clearance once they get into the blood. So there must be some optimal time points to collect the blood to get the maximum amount of the biomarker in the blood. And so far, based on our small animal study and also large animal study, it looks like we want to collect the blood as soon as possible. So if it's feasible, I would suggest collect the blood right after focused ultrasound as one of your time points. And then, of course, you can also have later time points.
Great. Thank you so much. Lauren, I want to ask you a question about some of the practical elements of doing these studies. And my question is, with focused ultrasound liquid biopsy, there's a couple of different, actually many different companies that are involved both on the FUS side and also on the liquid biopsy side. And so when you think about, again, a future here where this is showing promising sensitivity and specificity, how do you go about coordinating pivotal studies that may actually lead to practice change when you've got, neuronavigation guided FUS, MRI guided FUS, implanted FUS, and then you've got myriad companies that are doing this liquid biopsy on a commercial basis. How do you get any of these companies to either work together or compare different, platforms versus each other? I imagine that's quite a challenge, and I'd like to hear your experience and thoughts on that.
Oh, well, thank you. I mean, for better or for worse, fortunately, this is a kind of BloodPAC's specialty, if you will, that we have been, I think our focus has been on bringing together, again, all those stakeholders to work on developing projects, again, developing those standards and frameworks for the community, as well as, when it gets to a point of, trial design, helping in that area too. Our first step has been, obviously, was being approached by the Focused Ultrasound Foundation, which is wonderful and was incredibly beneficial to us in understanding the field more broadly. We have our goal over the next year is to bring together, again, numerous focused ultrasound companies to the table to help us understand their needs, their kind of the variables, the frameworks that they're setting standards in, pair that and kind of educate our assay developers on the liquid biopsy side about what their needs are and how they can work together and develop, again, projects that move forward with the community as a whole. So again, the project that we first established for the work within our community right now is that you're going to be leading, which is very exciting, is focused on establishing those pre-analytical variables. I will say that BloodPAC, again, focuses in 3 different areas, setting standards and frameworks, but we also have in the second branch of our tree, our data, BloodPAC Data Commons, where we aggregate data that supports the establishment of those frameworks and standards. The third branch is accessibility, which we can happy to talk about later. But I do think the big point here is our data commons that does collect data to support those frameworks. So while we have in this process of bringing together these diverse group of stakeholders to work on projects that they define that would benefit the entire community, the idea is to also continuously aggregate and have data submissions from those companies that would support the frameworks that we're actually creating. And then our members have access to all of that data, and we do provide access broadly, open that data up to the general public. I'm happy to share our data, the public portal as well as for members, our private portal for the data that comes in. But I do think it's kind of that pairing of bringing everyone together in the community to define the problems, kind of the easy wins, not easy, it's still hard, but the easier wins together up front, and then move progressively through the process. And that collection of data helps support the projects that we've come across, we've started initially, like the collection of pre-analytical variables and standardization of those that will be required ultimately for the submission of data to the BloodPAC Data Commons.
Thanks so much, Lauren. Patrick, I want to kind of build off of that and think about clinical studies to look at the utility of liquid biopsy. And I think you brought up the very important question of disease monitoring and potentially using this modality to help us differentiate between tumor progression and treatment-related changes. How do you see us actually designing? What is the optimal study design to be able to start to answer those questions? It's a hard question, I know, but interested in your thoughts.
Yeah, I think related to what Graham had said earlier, we need to start thinking about incorporating these blood biomarkers into trials. Right now, if you look across the field, unlike systemic cancers, so few trials actually even collect the blood or the spinal fluid to do this. And I think that's an important first step. I think the other thing that requires a change in mindset in the field is that there has always been an aversion to collecting spinal fluid. In some patients, it's thought to be too dangerous because the tumor is big and there could be issues of herniation. And certainly for those patients, you shouldn't be collecting spinal fluid. But I think for the large majority of brain tumor patients, it's actually safe to collect spinal fluid. And I think it would be important to incorporate more of those studies into trials. I think if we can do this like systemic cancers do in their studies, we'll start to collect the information to look at the utility of these approaches.
Thank you very much. With that CSF question in mind, Hong, could you talk a little bit about any preclinical work you've done in looking at CSF and whether focused ultrasound may have a role in increasing the yield there as well?
Yeah, that's definitely after we have some preclinical study to show we can use focused ultrasound to enhance the biomarker release into the blood. Then the next natural question to ask is how about CSF? So we don't have a concrete answer yet, so we just perform some preliminary study by collecting the CSF in mice, and then we do have evidence to show focused ultrasound can also enhance biomarkers release into the CSF, but stay tuned, more data will come.
Very excited to see that. So there's a couple of additional questions from the audience, and anyone on the panel can feel free to take a stab at these. The first is there any development in the downstream analysis of liquid biopsy assays using machine learning or complex regression algorithms to help detection of biomarkers? Thoughts?
I'll just give my initial thought. I think that's the goal. Part of the challenge is right now we don't have enough samples, so we're working on collecting that. For those kind of algorithms to where you need lots and lots of samples to work with. And lots of sample with ground truth, as we talked, that's a major challenge in some of our studies where in our initial study, it wasn't an inclusion criteria to have patients with tissue samples present. So we would have our blood samples but have nothing to compare with. So that was one of the challenges.
Any other thoughts on that question?
I have a question. So I'm just wondering, so far there's already many liquid biopsy clinical trials, especially, for example, for monitoring disease. Are there lessons that we can learn from those trials so that we don't have to make many mistakes before we reach the end goal?
Yeah, so I think you're referring to studies in other solid tumors, potentially?
Yes.
Yeah, I think it's a great point. You know, in routine practice already for tumors like lung cancer, which is sort of the quintessential example of liquid biopsy in the clinic, there are multiple ways that it's used. One is for actual de novo detection of mutations at diagnosis. So if somebody has a lung cancer that's been biopsied or resected, the clinicians will often obtain a liquid biopsy along with that and sometimes find key mutations, sometimes important targetable mutations, including EGFR. And then those are often checked at the time of radiographic progression again to see if that particular variant, for the classic case again would be like a T790M resistance mutation in EGFR mutant lung cancer, is that mutation variant allele fraction in the plasma increasing over time in response to a targeted therapy against that. And there are other examples, BRAF mutant melanoma. And so one of the challenges in brain tumors is we don't often have a key driver mutation like that that's really the one oncogenic switch that's driving the disease. And so it's not going to be as easy. I think one of the things we can do, as you guys have done in your preclinical work, is look at mutations that are common in this disease, things like TERT promoter mutations, and particularly the ones that are conserved throughout the course of the disease. And that's a challenge, but I think it's got to be a priority as we do these studies. There's one other question, and anyone can take this. How close to the tumor does the sonication have to be for targeted, for optimizing the ctDNA yield? So how close to the tumor on imaging should we be putting the ultrasound?
So that's also a great question. So one of the strengths of sonobiopsy is we can spatially target at a specific tumor areas. So we would hope that we can pinpoint specific brain area and then perform genetic analysis or molecular analysis for that specific location. It's like a spatial targeted molecular analysis, but do it in a non-invasive way. So I would say we want to target it specifically at the brain tumor region that you are interested to collect the molecular information from.
I have a follow-up question.
Yeah.
So I wonder, I mean, it's not something I can conceive studying this in clinical patients, but do you think it's necessary actually to open the blood-brain barrier for the same effect to happen? I know there is a study that was shown, I think it's called BrainFo, where they're just increasing the temperature using HIFU and then collecting samples to see if there's any change there. Do you think it's absolutely necessary for blood-brain barrier opening to actually occur for sonobiopsy?
That's a great question. I was just talking with Dr. Frank about ultrasound. When ultrasound applied to the brain, to the cells may release extracellular vesicle and release other markers. So I definitely believe opening blood-brain barrier is just one way that we can make sonobiopsy to work. And then if maybe we can also integrate with other modality like hyperthermia or mild heating or other non-microbubble-based ultrasound mechanical effect to enhance the release of the cargo from the cells to the interstitial space, and then combined with opening blood-brain barrier to release those biomarkers into the blood. But the point is, I do believe we have a lot of biological questions that we want to study and then make this technology work more efficiently.
So we have just a couple minutes left, and I want to just think about one other question for Dr. Woodward. So I know that you use focused ultrasound in your practice for a variety of therapeutic modalities as well. And I'm just curious about your experience and how patients go through this, what they think of it, how taxing it is on them, and then thinking about taking that situation and applying it to a very sick patient with glioblastoma who may have quite limited life expectancy, comes to the doctor a lot already. How do you see this kind of working into our patient population, if this looks promising?
Yeah, I think there's a number of ways to think about how we would use this sort of liquid or sonobiopsy data over the course of managing a patient. But the way that we're thinking about it right now is that the focused ultrasound component of the MRI scan just becomes another sequence in the MRI scan. So you get a T2 weighted MRI, you get a diffusion weighted MRI, you get an SWI sequence, you get a focused ultrasound sequence. And it just happens that before and after the focused ultrasound sequence, you draw their blood. And that becomes just part of the workflow. And so if we do it that way, then now we're correlating what's happening in the blood before and after focused ultrasound with the new imaging data. Again, getting a deeper level of structure and functional data so that we can better understand the disease. I think that's where we need to be going. And even if we don't get to the level of being able to formally diagnose the tumor with liquid biopsy, at least we'll have some functional information about the state of the disease. And that's when you get into the idea of, well, are we looking at this from a perspective of understanding a specific process or a biological pathway and then trying to assess that pathway in the blood somehow or the CSF? Or are we reverse engineering this from a pattern recognition state where we have a more broad analytical assay or process, and then we're going to then correlate that to either some ground truth on histology or a better understanding of the biology from the MRI, whether that's PET MRI or whether that's just a standard MRI.
And how much time roughly does that add on to their MRI, that extra ultrasound sequence, so to speak?
Yeah, and I think that's the other question is time and pain for the patient and discomfort for the patient. So with the evolution of a shaveless and frameless focused ultrasound MRI, which is where we are now, you can basically put the patient, they have slightly different head fixation, than they would in just a coil or a standard MRI. And the total time to, they have an IV for their gadolinium-enhanced component anyway, so you're doing the infusion or the bolus of the microbles at that time. And so it won't take more than 20 minutes, I think, to do a focused biopsy in an area. Now, if you wanted to sample a broader region, as Hong mentioned, and really do more of a broader molecular screening of an entire region around a tumor, that would take a little more time. But if you really want to probe a specific area of high metabolic activity or high CBV on the MRI, you could do that in a relatively short period of time.
Great. Well, thanks everyone very much. I want to thank each of our panelists for their participation and for the audience for listening. I think this was a very helpful panel, and look forward to continued collaborations with everyone in this space. Thank you.